Basin, and the New York City metropolitan area. In addition, climate change, like most water bodies around the world, is impacting the Long Island Sound as well, with warming temperatures and shifting precipitation patterns. And these warming temperatures, alongside the eutrophication of the watershed, contribute to harmful algal blooms that eventually lead to hypoxia and manganese reduction due to their microbial decomposition. One thing to note, however, is that in recent years, the Long Island Sound Partnership has made significant headway in reducing the nitrogen load of the sound, as well as decreasing the hypoxic extent of the sound, which we'll look at here. But to begin, we'll talk about my research question, which is how is decreasing hypoxia and pollution impacting Long Island Sound's demersal fish? We can see here in this graph, with the 5-year running average of hypoxia extent, a decreasing trend. However, it's very important to note that there is significant interannual variation in hypoxia, mainly related to the interannual variation in both warming and precipitation. To answer my research question on how hypoxia, or decreasing hypoxia, is impacting demersal fish in the Long Island Sound, I will use otolith age growth and microchemistry data, collecting otolith length data and using Campana's biological intercept method to back calculate length at age throughout each fish's life history, as well as measuring the incorporation of various trace elements that are controlled via both physiological and environmental controls. And in this figure at the bottom of the slide, you can see a brief outline of how I would go through and do these back calculations. We have a red X indicating the location of the core, or the primordium of the otolith, followed by annuli subsequent in black dots, and each of those annuli would just represent an individual distance over the time of the otolith and representing an annual increment of growth. To just review some of the objectives for my otolith microchemistry research today, we're going to look at magnesium and phosphorus. Magnesium is typically associated with somatic growth, whereas phosphorus is going to be related to overall growth in most circumstances. So those are going to be very important for once we get to parsing our annual increments later on. We will also look at manganese, which is our hypoxia proxy due to its reduction during hypoxic conditions in the Long Island Sound. And we will also look at strontium and barium, which are two salinity proxies. High concentrations of strontium are associated with high salinity, and high concentrations of barium are associated with lower salinities. There are also some additional elements that are not included in my talk today that will also be included in my eventual thesis, but just for the sake of time, they won't be included in our talk today. The focus of the talk today is looking at the age-length validations and annual parsing of the microchemistry data, and the elements that I just mentioned will be crucial to actually doing that annual parsing. And the parsing that we do look at today will also allude to some of the classifications that may result from the annual parsing of the microchemistry data. The main classifications that I'm looking to do are for the migratory contingent of each species, whether or not they are resident to the estuary or they're migrating in and out of the estuary, as well as looking at their mean annual hypoxia exposure, creating 3 different groups for relative hypoxia exposure: high relative, moderate relative, and low relative hypoxia exposure. And then subsequently looking at all the age length data that I collected across these classifications to look at how both hypoxia and migratory contingency is impacting their growth rates over time in the Long Island Sound. And just to note about our tautog and winter flounder, and we're going to be looking at later on in the presentation, we'll look at a single individual out of the total 60 tautog that I've analyzed, as well as 1 out of the 49 winter flounder that I will analyze. I've only analyzed about half of them so far. We're just going to do a quick zoom in to where our tautog for today was captured. Oops, a little bit too fast. So we're on the north central portion of the Long Island Sound, right next to the New Haven Harbor. And when we look at our magnesium proxy for somatic growth, we see a very distinct peak in the core, and subsequent to that, we see a minima co-occurring with the visual annulus, indicating that minima in our magnesium are going to indicate the occurrence of an annulus. And we see fairly good resolution on the dorsal axes, which is that extended axis on the right, your right side of the screen, and the ventral axes we see a bit more compression, and those annuli are a little bit less resolved within the magnesium data, but you can cross-compare across the dorsal and ventral axes and locate the minima within the microchemistry data. Moving on to phosphorus, which is our proxy for overall growth. Rather than seeing a consistent peak in the core, we actually see a bit of a dip in the core followed by a peak before their first year of growth. And then we see a slight minima following that peak. Whoops, I am not good with this computer, sorry. After that first year, we see a very distinct decrease in growth, especially on this compressed ventral axis, but we do actually see a slight peak on the dorsal axes, indicating just higher growth period in the summer for this fish. Once we get to the third, fourth, and fifth years along the dorsal axes, the third year is a bit hard to resolve. This could be related to the incorporation of reproductive growth in this individual, so in our magnesium where it's just somatic growth, and these fish typically reach sexual maturity around 2 to 3 years old, so there could be something occurring around that time period that's causing a bit of difficulty in resolving that third annulus in the phosphorus data. In looking at our strontium for this individual, we see a very distinct low period in the core and in the first year of life, followed by an offshore migration, or an offshore migration in the winter. Then, in the spring of their first year, or the spring that indicates their first year, they migrate back inshore, and that's represented by this decrease in strontium in the summer period when they're in the estuary feeding and also reproducing. But at that point in this summer, this is a 1-year-old fish, it's a bit too young to actually reproduce. So we do actually see them making migrations before their sexual maturity, which isn't well documented in this species. In terms of our barium, we see a inverse pattern, at least in the first 2 years of life, with minima occurring or co-occurring with each annulus. However, once we get to the third, fourth, and fifth years of life, we actually see peaks in barium. I'm not exactly sure what's happening here, but once I actually get to annually parse this data and do some more robust statistical analyses of these annual increments and the annual microchemistry data, I might have a more conclusive answer to why barium is starting to co-occur later in life in this species with strontium. Moving on to our hypoxia proxy in this individual, we do not see a very distinct signature in the core indicating that during its spawning or its mother was during its spawning there was no hypoxia present in the water column, as well as the mother of the individual was likely not exposed to hypoxia during the development of the eggs. However, we do see a minimum occur in the first spring, which is a bit surprising because in the spring and summer is typically when you're going to have hypoxia occur in the Long Island Sound. So this could just be indicating that during that first year of life, there was no there was not a strong strong algal blooms. There was colder temperatures and potentially lower precipitation, leading to lower rates of eutrophication and hypoxia. However, in this individual, we do see significant interannual variation. So in the second year, we see a maxima indicating high hypoxia during that annual year. And in the third year, we see a minima, fourth year, a maxima, and in the fifth year, we see another minima. So very significant interannual variation in this hypoxia proxy, and we see the same pattern both on the dorsal and the ventral axes. And this proxy is our strontium and our magnesium ratio. This is an alternative proxy. However, it's very well correlated with each of our visual annuli. With our first here, second, third, fourth, and fifth, we get really good annual resolution. This is pretty consistent across all individuals that I've been able to look at so far. So our the strontium-magnesium ratio is going to be really important for once I actually get to parse up these data annually. Moving on to our second individual, our winter flounder, just zooming in once again to the capture location, fairly similar location, but just a bit further south in the sound. In winter flounder, we see a much less distinct peak in the core. This could just be due to this individual and me actually just not polishing it very well and not actually getting to the core. But it's hard to say. There are some individuals that have a very distinct core signature, but this individual not quite as much. But we do see that same minima occurring or co-occurring with each visual annuli, indicating winter formation, because winter flounder are spawned in the winter, hence their name. In the second year, again, we see that same minima co-occurring with the second annulus, very close to that outer edge, and just a little bit short period of growth before these fish were caught in the spring. In terms of phosphorus, it's a lot messier. The peak in the core is actually even less well resolved. It's very low concentration relative to the peaks in the summer growth periods later on in life, but we are still able to resolve some annual increments here. However, there's a bit of weirdness going on, and again, similar to the previous example on the tautog, I think some of the weirdness that occurs, at least in the first to second year of life, is related to their sexual development and reaching sexual maturity. So we see a bit more resolved pattern in magnesium, but when we look at phosphorus, we get a little bit more noise due to that incorporation of reproductive growth. In looking at the manganese proxy, we do see a bit of a core signature, but nothing too significant, indicating that there's no hypoxia when this fish was born and in its first few weeks of life. However, the minima co-occur with our strontium, so that's sort of indicating that these fish are offshore or in more saline waters in that first year of life. Once they're in the winter, they're in the winter period. And then we have a very distinct manganese peak within the first and second year, between the first and second year in the summer, indicating a year of high hypoxia. And after that second year, we also see the beginnings of an additional peak in hypoxia. Moving on to our strontium proxy, we see again a very low signature at the core, indicating that these fish were spawned very far inshore, likely near the mouth of a river, followed by a peak in strontium right around that first winter annulus, and then followed by a decrease and a subsequent increase. So we have the co-occurrence of annuli in strontium with the maxima in strontium. In barium, we see, again, not a very distinct core signature, so maybe this fish was not spawned inshore because of that low signature, but it's a bit hard to tell at this point or for this individual. But we do see a very distinct peak at the second annulus, and the first annulus we see a very distinct minima, and at the second annulus we see a very distinct peak. Not exactly sure what's going on there, but there's some very interesting migratory variability within winter flounder. They display metapopulation characteristics, so they are constantly moving around. It's hard to predict exactly what they're going to do. So we'll see once we actually get through the annual parsing and have some more robust data to talk about. But we do see a very similar pattern in the strontium-magnesium ratio as we did with the tautog, where the peaks in the strontium-magnesium ratio are co-occurring with each annulus, again, meaning that it will be very helpful for actually breaking down these annual increments. So generally, the next steps are, as I've emphasized a few times, to parse the annual increments for all individuals. This is just an example for each species. And then I can evaluate tautog and winter flounder migration patterns, try and resolve some of the weirdness going on with winter flounder, and see if there's any variation within tautog. They typically have very strict migration patterns, moving inshore in the spring and summer and offshore in the winter. And Once I have those two groupings, I can also look at hypoxia exposure and look at the overall impact of their migration and exposure to hypoxia on growth. And just some last minute acknowledgements. I wanted to thank Kirk Gottschall and CT Deep for providing the samples. I got all these samples from the spring 2024 Long Island Sound Trawl Survey. So big shout out to them. The Limburg lab and my whole lab cohort for helping me throughout this whole process. Deb Driscoll and the Analytical and Technical Services Department at ESF for helping to facilitate my research and actually allowing me to collect all my data. And that's all I have. So, yeah.